Cryptomonads. Their name literally meaning hidden single cells are among the most inconspicuous of the algae. There are several reasons for this. Cryptomonads are relatively small, 3 to 50 micrometers in length members of the phytoplankton. They are often most abundant in cold or deep waters. They are readily eaten by a wide variety of planktonic herbivores, and natural collections are not easily preserved, the cells tending to burst readily when subjected to environmental shock. Cryptomonads are probably the most appreciated by plankton ecologists who recognize their high quality as food for zooplankton. Cryptomonas mersoni. The cells were approximately symmetrical with rounded antepex and ventral view under LM, and contain 2 chloroplasts per cell without pyrenoids. They show the sigmoid shape with a sharply pointed and curved antepex in lateral view. Cryptomonas ovata are known to double their populations in less than a day. Higher rates should be expected for species with smaller body sizes. This is a freshwater species and found among vegetation. Measurements for these species could be 20 to 25 micrometers. Cryptomonas pyrenoidifera. This is a brownish freshwater species. Cryptomonas erosa. This is a freshwater species. It doubles its population in less than a day. Higher rates should be expected for species with smaller body sizes. Chelomonas. Organisms under Chelomonas are colorless due to the absence of a chloroplast and pyrenoid. Cytopharynx is deep, low, and half surrounded by granules. One contractile vacuole is anteriorly positioned. Chylomonas paramecium. C. paramecium cell size is 8 to 40 micrometers long and 4 to 15 micrometers wide. Posteriorly narrowed, slightly bent dorsally, widely distributed in stagnant water. Leucocryptus marina. The morphology of L. marina is a rigid cell with asymmetrical droplet shape that is 7 to 30 micrometers long and 3 to 10 micrometers wide, with 2 rows of large extrasomes and 2 approximately equal flagella. This species is a solitary life form and is widely distributed and feeds on nanoplankton. Chylomonas oblonga. C. oblonga is a solitary life form and has a morphology of a typical Chylomonas wherein it does not contain a chloroplast nor a pyrenoid, resulting in its colorlessness. This species has 2 flagella of unequal length. Goniomonas is colorless and does not contain a plastid. Food organisms are taken up by an anterior tubular invagination, the infundibulum, and digested in food vacuoles in the cytoplasm. Storage granules occur inside an extension of the outer membrane of the nuclear envelope. Large ejectosomes occur under the anterior plasma membrane and small ejectosomes occur between the periplast plates. Goniomonas truncata. Cells flattened, outline tending to ovate, rounded posteriorly, obliquely truncated anteriorly. 2 nearly identical flagella, about half as long as the cell length, emerge from the small anterolateral depression. 4 to 6 densely packed or fractile extrasomes located parallel to the anterior margin of the cell. Nucleus usually situated mid-laterally to the side where flagella emerge. Contractile vacuole lateral at the opposite side of the cell. In some cells, 2 to 4 more or less conspicuous, slightly oblique longitudinal stripes present on the periplast. In other cells, no stripes visible. Sometimes, swimming with flagella directed forward. Sometimes in contact with substrate, either motionless or gliding with one flagellum trailing. Goniomonas amphinema. 2 unequal flagella with different orientations, the long one which trails behind and the short one which is held straight while swimming. It has flagellar appendages located only on its short flagellum. A row of extrasomes parallel to the truncate anterior margin. The cell size of this species ranges from 4 to 8 micrometers. They are flattened and oval-like in shape, however, most of this species observed being obtuse anteriorly and rounded posteriorly. The granular area on the apical surface is carbohydrate-rich, containing GLC-NAC residues. Lining on the surface is defined as delicate stripes, are actually deep grooves running down the length of the cell. Goniomonas avonlea. A colorless bactivorous marine flagellate with flattened cell body. Cell size ranges from 8 to 11 micrometers long, 6 to 7 micrometers wide, somewhat truncated at the anterior. 2 subequal flagella are slightly longer than the cell and arise from an anterior depression. One flagellum is directed forward, whereas the other runs backward toward the cell posterior through a longitudinal groove. A conspicuous band of large ejectosomes run across the anterior margin. Younger cells tend to have more pointed posterior end compared to older cells, which have a more rounded end. When swimming, the cell rotates around its longitudinal axis. Swimming cells slide along surfaces with the posterior flagellum trailing beneath the cell. It has a distinct 18S rRNA gene from those of other Goniomonas species. Goniomonas pacifica. Cells are 4 to 10 microns long with several distinct longitudinal ridges on both sides of the cell. There is a row of about 7 to 9 ejectosomes near the anterior end of the cell, which is truncated, with the posterior end rounded. 2 flagella of similar length emerge from a small anterior depression and are directed anteriorly. When the cells are swimming, 2 flagella diverge in different directions. Cryptomonads is a group of species described by Anton Hanskerg. They are generally barrel-shaped but slightly compressed, the ventral surface being somewhat flattened. Majority of them are blue-green in color, which is sourced from the bilirubin CR phycocyanin 645. The cells have a strong tendency to alternate between a motile state and a palminoid state, in which they produce macroscopic mucilaginous aggregations. They appear to be the only cryptomonad genus to possess an eye spot. However, it is not present in all species. Cryptomonas mesostigmatica. This species possesses a red eye spot in the center of the cell, which is an extension of the chloroplast beyond the pyrenoid. It has an overall blue-green color, whereas the eye spot itself is pale yellow and is situated deep within the cell, between 1/3 and 2/3 of the way from the anterior, flagellated end. Between the eye spot and the cell surface are 2 vacuoles. The small vacuole is positioned close to the eye spot disc, whereas the larger vacuole lies immediately adjacent to it. Cryptomonas nordstedtii. It is described as having subspherical or oblong cylindrical cells, 6 to 8 micrometers broad and 9 to 12 micrometers long. The cells have 2 anterior subsequal, briskly moving flagella which emerge through the anterior vestibulum. The chloroplasts are simple, parietal, and of a color between that of a cankered brass and blue, and the pyrenoids are spherical. Cryptomonas salina. This species has a broad anterior, a subapical opening of the gullet on the flattened ventral side, and a tapering posterior. It has an arrangement of numerous small rectangular plates of varying sizes in different parts of the cell, specifically in longitudinal rows with the anterior raised edge of each plate out of step with those of neighboring rows by a constant amount. The protrusions of each individual trichocyst gives a markedly serrated appearance to the periplast sections, and they're situated beneath the anterior-facing edges of each of the plates. This consequently gives the periplast a clearly hexagonal honeycomb pattern which overrides the spiral aspect of the arrangement. Cryptomonas bachmanii. Cells are barrel-shaped to ovoid, 10 to 18 micrometers long and 8 to 13 micrometers wide, and blue-green in color. A single chloroplast and a massive pyrenoid are present in the cell. They have a gullet but lack a furrow. Each cell has 1 massive blue-green chloroplast with a large pyrenoid. Also, a large, prominent contractile vacuole is located in the anterior of the cell, which is specialized for capturing uncontaining bacterial cells. Numerous large starch grains are usually present in the cell. Cryptomonas cerulea. Cells are ellipsoid and sometimes slightly concave dorsiventrally, measuring 8 to 12 micrometers in length and 4 to 6 micrometers in width. The cell posterior is rounded. Inner and surface periplast components consist of small rectangular periplast plates. A single blue-green chloroplast is present, with a single pyrenoid and a prominent stigma, which is associated laterally with the pyrenoid. Only a vestibulum and a gullet are present. Flagella are shorter than the cell length. Cells often form extensive mucilage in which groups of cells remain embedded and attached to a substrate. Ubiquitous in Rocky Mountain lakes, reservoirs, and streams.